Flash, duck, and cover, and do it fast.
See that you help by removing all of the litter, junk, old newspapers, and trash.
Rapid climate change in the polar regions is outpacing the ability of ecosystems to adjust. This disequilibrium affects more than the environment. Its wildlife suffers the consequences in ways only adaptation can aid them, to an extent. The timing of seasonal events phenology is shifting. Penguins can't reproduce at the same time they used to. With biotic and abiotic factors constantly changing, they have to modify their inner clocks to compensate for this mismatch. This affects penguin breeding seasons, creating interspecies competition over the same area of land. They are now experiencing one of the largest trophic, temporal, and population mismatches in ecology. So we have to turn the gears before it's too late. Research communities, like oceanites.org, keep track of these patterns to inform decision makers with evidence to safeguard the Antarctic. Donate today, because when they fall out of sync, so will their survival.
Huatambo, a peninsula on Argentina's coast, is home to one of the largest colonies of Magellanic penguins. While this group seems abundant at first glance, the breeding population has declined by over 50% since 1982. Each year, penguins stay on land for 2 to 3 weeks to shed all of their old feathers and regrow new ones in a process known as the catastrophic molt. During this time, molting penguins lose insulation from their feathers and refrain from hunting for food due to the cold ocean water. Climate changes cause an uptick in extreme weather events such as heat waves and extreme rainfall. When penguins are less insulated during the molting season, they struggle with thermoregulation and are susceptible to death from the unusual weather patterns. It is up to us to support conservation efforts to save the penguins from the consequences of climate change. Join the cause by donating to the IUCN today.
Around 70% of the penguin population has decreased in the last 75 years. One of the most vulnerable times of a penguin's life includes the catastrophic molt. For 2 to 5 weeks, they lose all of their feathers and grow new ones while unable to catch fish without quickly freezing in the cold water. They rely on fat reserves and often die of starvation without sufficient foraging before the molt. Protecting their habitats is vital, but the help of federally funded organizations has been a pillar of scientific prosperity. The Trump administration plans to defund almost $4 billion previously allocated funds for NIH, NIST, NSF, and additional organizations that provide maintenance, research, restoration, conservation of land, and preservation in vital ecological sites. These historic cuts are preceded by canceling or suspending almost 8,000 grants, while federal science agencies have lost over 20% of their staff. It is more important than ever to stay informed, be active in local governments, and vote. Protect science before we lose what makes our world worth living in.
Eustachian tubes and the throat are lined with a delicate membrane. If, under a microscope, you look at the lining of the nose...
The old man gorilla may reach nearly 6 feet in height. He may weigh 500 pounds.
On the coast of South Africa, among the rocks and sand, lives an African penguin colony. Breeding season has begun. Among the crowd, a pair reunites. He carefully settles everything: the branch, the spot, the perfect burrow carved into the cold earth to survive the intense sun. Soon after, she lays an egg. Protection of this fragile, vulnerable, small piece of life is now of the utmost importance. Some days, she goes to search for food while he huddles in the nest to incubate their egg. Other days, the roles switch. Today, he is preparing to go out to sea to hunt sardines and anchovies. He calls out one more time to her, then disappears into the water. She waits and waits throughout countless days. The sun is getting hotter than usual. She cannot leave, not yet. But he never returns. She now has to make an impossible decision. With scarce food and unbearable sun, it's her duty to find resources, leaving her egg completely alone. But duty calls. Slowly, she emerges from the nest. In the open, the world is harsher, and the burrow's only protection is gone. She successfully cools down and gathers some food, rushing to the nest before any nearby predators catch her. In the distance, shadows move. Predators. Drawn by opportunity. She stands her ground, weakened and exposed. Yet resilient enough to survive. And still, everything was lost. The breeding cycle repeats for the rest of the brood. But something is changing. African penguins are suffering from climate change. Longer journeys, hotter days, unprotected nests, fewer chances. In a burning sky, even the most resilient can't win.
1,000 kilometers from the coast of Ecuador lies a remote set of islands. Shifting landscapes and equator-bound terrain provide an abundance of opportunities for diverse ecosystems with highly specialized individuals. These are the Galapagos Islands. Without human disturbance, life exploded in the Galapagos. As a result, 60% of the species here are endemic to this particular piece of the globe. Once geographically isolated from the rest of the world, these islands exhibit species with highly specific adaptations. The Galapagos flightless cormorant is just as its name describes, distinctly flightless. Dark, shiny feathers and bright blue eyes give the species a distinguishable appearance. These birds can weigh twice as much as their flying counterparts, but have tiny, stunted wings. With an additional loss of flight feathers over time, Galapagos cormorants are the only species of cormorant that is incapable of flying. Trading the skies for the seafloor, heavy bodies and small wings allow these birds to dive deep into the coastal sea. Galapagos flightless cormorants can dive up to 73 meters to hunt for fish, eels, squid, and octopus. Their distance from the Ecuadorian coast renders these birds isolated to the rocky shores of the Galapagos. April has arrived, and these cormorants are ready to find partners. Courtship begins with a water dance. Birds extend and flick their necks, swirling around each other in the sea. Females outnumber males, so she holds the power of final say for a mate. She will choose a male that shows strength in both foraging and parental care. Lucky for this male, she likes what she sees. This male offers seaweed he has foraged off the seafloor to his nesting female. This pair will build a ground nest together from found gathered materials. These monogamous birds will take turns incubating the growing chicks, switching off to forage. Since these birds cannot fly, they gather together to nest in groups on rocks close to the water. With advancements in travel technologies, the Galapagos Islands are no longer inaccessible to humans. Tourism has introduced new threats to these birds that once lacked a natural predator. Invasive cats and rats scavenge along the shoreline, where this pair's eggs sit vulnerable and exposed. This male will occupy the nest, both protecting and incubating his 2 eggs, while his mate leaves to forage. What he doesn't know is that she might not come back. A new male courts this female. While individuals of flightless cormorants practice monogamy when caretaking, the species as a whole is polyandrous. This female could leave to mate with up to 3 other males per courting season. If she abandons her nesting mate, the outcome could benefit the species as a whole. If this female engages in courtship with multiple males in one season, she might be able to set up 9 potential hatching eggs, as opposed to just 2 or 3. This new partnership will increase the odds of successful reproduction and sustain healthy population sizes with more genetically diverse individuals. This male will finish the rest of the parental care on his own, hopefully succeeding in raising at least 1 healthy chick from his set of eggs. Pututumbo, Argentina, a rugged stretch of the Patagonian coast, is home to one of the world's largest gatherings of Magellanic penguins. At its peak, over 200,000 breeding pairs nested in burrows they dug, or under unfriendly thorny bushes protected from the sun and grazers, such as cows or guanacos. Researchers like Dr. Dee Boersma have spent decades documenting their lives as marine sentinels. It's late fall in Patagonia. The wind is biting. Once a year, the Magellanic penguin loses all of its feathers and grows new ones in a catastrophic molt.
3 penguins in different stages of the molt, all adults.
Penguins are the only species to drop all their feathers and grow new ones in a couple of weeks. A biological overhaul replacing every single feather is one intense event. Most birds drop one feather at a time, or replace a few feathers at a time throughout the year so they can keep flying, staying warm, and conserving energy.
Kill gulls over the cormorant colony.
But penguins rely on a perfectly sealed waterproof suit to survive the frigid South Atlantic. They cannot afford a single leak. Before their annual molt, Magellanic penguins undergo an eating phase. A penguin that usually weighs around 4 kilograms may gorge itself until it hits a staggering 6 to 9 kilograms. If they don't reach this peak mass, they won't have the energy reserves to grow new feathers. Recent research into penguin physiology shows that individuals can lose up to 45% of their total body mass during this fast. If they start too thin, they face the very real threat of starvation before the first new feathers even break the skin. Magellanic penguins are opportunistic foragers. They hunt for the oily Argentine anchovy. Further south, they switch to the Chilean sprat. They are efficient predators, capable of consuming 5 grams of fish any chance they get. These are the proteins and fats necessary to power a long fast. This is a season of atmospheric variations. In Patagonia, you wear layers because the morning is freezing, but by noon, the sun is scorching. But the penguins have no layers. As the new feathers begin to grow underneath the skin, they push the old feathers outward. For a brief window, the penguin is covered in a double coat. Feathers create a thermal challenge. The energy required to grow an entire plumage is incredibly high, generating massive amounts of internal heat. Trapped under a double layer of feathers and unable to enter the water to cool off, the penguins are at a constant risk of overheating. The unfeathered patches of skin around their eyes and bills turn a bright, hot pink because blood is just under the surface of the skin. These are the areas where the body sends hot blood to the surface to be cooled off by the wind. It is a grueling 19-day test of endurance. A penguin preens for hours using their bills to spread waterproof oil from a specialized gland, the uropygeal gland, across their feathers. This allows them to stay bone dry and warm while diving. But as they molt, they stop oiling. Without that protective coating, their feathers become brittle, dull, and lose their deep black luster, taking on a worn-out, brownish hue. Finally, the last of the old, ragged patches fall away, usually from the head and the flippers last. The transformation is complete. The old, worn-out plumage is gone, replaced by a sleek, waterproof tuxedo with the characteristic black and white bands on the chest. Their weight has dropped, sometimes as low as 2 kilograms, leaving them lean and hungry. They are finally free. They head back into the surf to begin their long winter migration, rarely touching land again until they return here in September. They have survived the catastrophic molt.
The spotted hyena, a creature with a dynamic social culture comparable to primates and dolphins, with extremely strict hierarchy rules. Although it is common to praise the king of the jungle and look down on these harsh scavengers, there may be more to these beasts than meets the eye. Hyenas are often depicted as simple, dim-witted creatures. Despite this misconception, hyenas cooperate extremely well with their clanmates, only needing to form a small group that can hunt down even the most formidable prey, with inevitable demise. While this hunting strategy allows for difficult prey to be taken down, females dominate the eating portion of the process. Spotted hyenas have a matriarchal social structure where ranks are passed down by mothers. Although male cubs are protected from this nepotistic hierarchy while young, their rank will soon be knocked below all the females and cubs of the clan. Females are far more aggressive than males, ensuring they get more than their fair share of meals. This male is always left with bare bones, or nothing at all after expending energy on a group hunt with females. To avoid this, he must find food alone, and this isn't always easy. He is forced to sulk past the larger prey that isn't worth the trouble while alone. He finally smells something edible just on the other side of a bush, but it's already taken. A pack of African wild dogs devour their recent kill in front of him as his stomach grumbles on the outskirts of the clearing. These African wild dogs are well aware that this hyena is not one of them, and they are not fond of their food being watched by an outsider. He is forced to find another way to get a meal. While he may have been hoping his dog-like face would be enough to get a bite of that meal, he's not closely related to these African dogs at all. The Hyenidae family that hyenas belong to shares the same subclass as Felidae, which contains felines. Their closest ancestor to dogs branched off into Feliformia and Caniformia around 42 million years ago. This puts hyenas in a distant, bone-crunching family of their own, with specialized intelligence and social cooperation to show for it. While this hyena has the teeth to smash bones, these low-ranking gnashers experience a high risk of tooth wear and decay from eating leftover bones. He expends substantial energy and tooth stability to break down the least nutritious parts left for him from a group kill. His next journey to find sustenance will be harder than the last. Although few skeletons can stand up to the pulverizing force hyenas bring to the table, he may be left with scraps again today. Luckily, hyenas evolved a digestive system built to handle a scavenging lifestyle. The low pH in hyena stomachs combined with their short digestive tract helps to kill pathogens and then quickly excrete them before they can populate inside the body. A hyena's life is hard, and infancy was no exception. As high as 60% of cubs don't survive the whelping process, and many are killed by siblings after being born with fully formed baby teeth and plenty of aggressive hormones. Although cubs lose these baby teeth and grow 33 to 35 adult teeth, they do not achieve their peak bite force for up to 5 years. The powerful jaws of an adult house enamel-coated teeth that can consistently withstand 1100 psi of bite force, but cubs rely on their mother's milk to sustain them for around 12 months as their jaws develop. Cubs have more bite force than the measly 160 psi that humans generally max out at, but they are at a huge disadvantage in the African desert. For a young male who is aging out of his rank just below his mother, there are about 8 months after being weaned before he will gain most of his bite force at 20 months old. He will be on his own from now on, but because most female hyenas refuse to mate with hyenas of their own clan, he will need to search for another clan to prove himself in without being torn apart. If he manages to join in at the bottom of the hierarchy, he will gain the chance for reproduction and die an early death. If a male cub can withstand his traumatic birth and then survive long enough to develop substantial jaw strength, he can spend his life fighting for a scrap of food in a neighboring clan and dream of the day he might be able to relax and enjoy a day or two in the hot African sun.
In this task, scientific photography is an important tool of analysis. Its techniques are the product of more than 90 years of development.
Deep within the forest, a small tree-dwelling mammal moves through the canopy. At first, it looks ordinary, until it gets dark. This mammal can fly. Bats are the only mammals capable of powered flight. This is the ability to generate lift through muscle contractions. Unlike birds, bat wings did not evolve from feathered arms. They evolved from hands.
The most feasible explanation is that the more distant ancestor of bats were small mammals that lived on trees, and over the course of their evolution they started evolving the ability to glide. So they evolved membranes that connected their arms with the rest of their body or their hind limbs, and that allowed them to sort of maybe pursue prey or move across the environment by bridging gaps between trees and foraging in the forest. And so we think that after acquiring that ability of gliding, then they evolve more sophisticated ways of movement by evolving membranes between their fingers, and then the elongation of the fingers, which then turn out to be the wings of bats, so they can flap, and then they're able to fly with much more maneuverability. Right now, if you look at the fossil record of bats, the first complete skeletons that appear in the fossil record, they already look very similar to modern bats. They already have very well-developed wings and other aspects of their anatomy that shows us that they were already able to fly. And scientists have looked at some structures in the ears of these bats, in these fossils, and to try to infer if they were able to echolocate.
Echolocation is a high-frequency biological sonar that transforms returning echoes into a map of their surroundings. They already dominated the skies, but to dominate the night, they had to see. And this technique gave them a brand new pair of eyes.
When we compare the anatomy of the structures of bats today that echolocate with species that might be closely related or they're somewhat sort of morphologically more standard within mammals, and then see what the difference are between those two. And we know that, for example, the middle ear ossicles of bats, they are tuned to vibrate with the high frequencies that bats produce. They have super fast muscles in their larynx to produce echolocation called pulses and things like that. So they have a lot of adaptations, but how each one of those evolved is something that still remains a big mystery, again, because the fossil record is just not very informative of that.
Their wings, echolocation, and ecological diversity continue to provide clues to biologists about adaptation and evolutionary possibilities within other mammals. Today, museums hope to preserve these bat specimens for our understanding of how animals evolve and continue to be an essential part of the ecosystem. From climbing trees to flying in the night, this is how bats became the dwellers of the sky.
This is the Burke. It's Washington State's oldest museum, home to more than 16 million artifacts and specimens spanning natural history, paleontology, and the indigenous art and culture of the Pacific Northwest. Behind these exhibits are world-class researchers using the museum's invaluable collections to make new discoveries about the world. Traditionally, unless you physically stepped inside, opened a scientific journal, or happened to stumble upon a video, you would rarely see the research happening at institutions like the Burke. That's where Chris Snyder comes in. Chris is the Burke Museum's digital media specialist, responsible for the museum's social media presence. From dinosaur reels to spider videos, he's the one bringing the Burke science to millions of people online. For Chris, social media isn't just about getting views. At its core, scientific communication is about making science feel human, accessible, and inspiring to people outside museum walls. That's especially important at places like the Burke, where a single exhibit can completely change the trajectory of someone's life.
Sharing curiosity-provoking stories about science or about the natural world, that is how new scientists are made, right? There's a reason why we have so many field trips of kids coming into the museum physically. Some of those are going to have their lives changed by, looking at a dinosaur tooth for the first time and say, this is what I want to do. I want to start asking questions about the natural world. The paleo people talk about this all the time. They're like, kids love it, and it's a gateway drug into other forms of science.
But in the digital age, traditional forms of scientific communication alone are no longer enough to reach modern audiences.
If you actually want to teach somebody how something works, I think reading is really good. I think long form lectures with visuals are really good, but maybe what this leaves out of the conversation is the question about where you're actually going to reach people. As much as ideally, we would all be learning about science through the printed word, lectures, so on. Social media has captured the attention of, millions upon millions of people, made our attention spans sort of not capable of consuming scientific communication in the classic way.
Today, Chris uses both short-form and long-form content to bring the Burke Museum's research to audiences around the world.
Social media is a tool for giving people a behind-the-scenes look into science that has never existed in the history of human time. Never before have you been able to film a video in a couple of hours, edit it, put it out, and reach genuinely millions of human beings all over the globe. I can just be like, this looks really cool, and the instant I put this in front of somebody, they're just gonna find it interesting and they're gonna watch it. So short form video is good for showing people things that they've never seen before and inspiring people's curiosity.
But social media comes with its own challenges.
Short form that reaches lots of people and gets people interested in stuff they didn't even know that they could be interested in, but doesn't, by nature, can't provide a ton of context, can't provide much complexity. Then you have content that is, medium form, 10 to 20 minutes, can go into things at a greater depth, but you're still limited in how complex of an idea you can communicate by the fact that you have to keep and maintain people's attention for that whole 20 minutes. Ideally, you're doing science communication in all the different forms for people who are interested at different levels.
One of the hardest parts of Chris's job is balancing scientific accuracy with audience attention.
I take it as a given that what I put on the internet has to be true. It's not necessarily a question of accuracy so much as it is how much accurate information can we put in this thing. And as a creator, I am always having to suppress the part of me that wants to add more information and more complexity. Experts really want to put the complexity, right? The way I see my job is trying to make sure that the show is entertaining so that people don't leave, because if they leave, then we can't tell them the cool thing.
And the job is only becoming more difficult. AI-generated videos and images now flood social media feeds with misinformation and fabricated content.
There are videos of things that never happened that are circulating out there, and people are viewing them and thinking maybe they did happen. Ideally, videos that you see on your phone that look like they're of real life should be of real life. And if you can't tell the difference between what's real and what's not, it's gonna be really difficult.
There has, understandably, been a lot of backlash against AI from the public. People want authentic content that they can trust. Interestingly though, Chris has also seen some more critical thinking from the public when engaging with content online.
Because people now know that things can be AI generated, it's generated a lot of skepticism among the general public about anything that they see, whether it's AI generated or not, because people know that they can't tell the difference, right? And so they are pretty quick to say that anything that doesn't quite seem right is AI generated. We run into this on the Burke's socials all the time, where people think that my voice is AI generated when I do like voiceovers and stuff like that. If a visual thing is too perfect looking, people assume that it's AI generated and click off, you know? I don't think that's a terrible thing. It's really frustrating when I make something that is real and then people, you know, accuse me of AI generating it, right? But on the other hand, it makes me feel like, you know, the public is being appropriately skeptical about what they see online. I would prefer that to the alternative of thinking that everything they see online is true.
At a time when misinformation and conspiracy theories spread faster than ever online, rebuilding public trust in science has become increasingly difficult. Chris believes social media can play an important role in repairing that relationship by making science feel more personal and human.
I'm sort of taking the really cool research that is happening in the collections, or just the expertise, the characters that are here. I mean, the people who do science, the people who've dedicated their lives to science, you know, particularly at museums, they don't make a ton of money. They're in this for the passion. I think a lot of people don't know that. So vertical video, for all of its flaws, puts a person you've never met in front of you, and if the video is done right, you get a sense of who that person is like that. You see their stoke for understanding how the world works. I think that that is really good for humanizing the work that happens back here. And I think when institutions have a good social media presence where people feel like they're kind of part of it, you know, they get to meet the characters involved and see how just like genuinely psyched the scientists are about the work that they're doing. There's something deeply human there that people can connect into, and I think it humanizes the whole process and builds trust.
In an age dominated by algorithms, misinformation, and shrinking attention spans, scientific institutions are being forced to rethink how they communicate with the public. And for museums like the Burke, social media may not just be a marketing tool, it may be one of the most important ways to inspire curiosity, humanize science, and rebuild public trust in the people behind the research.
I want to make sure I'm like not just, you know, saying words, and I'm actually like saying something meaningful here.
Mischievous tricksters, bad omens, pesky annoyances, intelligent messengers. Crows are highly personified creatures. With glossy black feathers, deep curious eyes, and a heavy tapered beak, the contrast of this fully black bird against the bright blue sky is immediately recognizable. In modern residential spaces where humans live, the American crow is more so currently depicted as something to shoo away. These birds navigate highly complex group dynamics. Families of crows will disperse and occupy specific foraging territories during the day. However, Separate groups of crows will resolve their previous conflicts with each other and continue to migrate back into their larger roosts, cooperating as individuals in greater communities. American crows are highly opportunistic scavengers with an unspecified diet that includes up to 600 different types of food. Crows have feeding options, like hunting for eggs of other passerines, pirating food from squirrels and other birds, and scavenging for discarded human wastes. This crow recognizes a human that feeds him. With accumulated growth of human populations, sparse dispersed homes with greater green spaces transition into densely packed cityscapes with significantly reduced natural plant agriculture. Urbanization of land that directly supports human lifestyles threatens the biodiversity of spaces in between. With big families to feed and limited nutritional food and water availability, these crows face a puzzling struggle of resilience. Cities get hot due to the high albedo of concrete, and when water gets scarce, crows find solutions, like sipping from this leaky public faucet. Males and females work together to foster successful nests. This young crow tears at an old cardboard box for nesting material. Younger crows within one family will actually delay breeding until they reach about 2 years in age in order to aid in parental and foraging care, operating as family units. After much careful testing, this crow finally chooses a stick for its nest. High social intelligence, in combination with their opportunistic, omnivorous diet and social learning skills, gives crows an enhanced ability to exploit resources to their advantage, despite the challenges that they're faced with. This group of crows drink and play in a Seattle Public Utilities reservoir. Sliding down slippery tarp, they problem solve by climbing up this yellow rope. Like other passerine birds, American crows utilize a fly and forage strategy. Crows will often fly far from roosting sites in a diurnal migration. They will forage in small familial groups of around 3 to 4 crows, often taking turns occupying the role of sentinel or lookout. Crows have learned how to use anthropogenic cues to better locate resources. With the introduction of more stimuli, cities offer more opportunities to form familiarity with local rewards and with local threats. The intelligence of the modern American crow allows this species to make assessments of human resources more rapidly and use areas of high resource abundance more readily, like these open garbage cans. These birds live in a proximal, long-term association with humans and actually benefit from the food and shelter that humans provide without affecting people positively or negatively. This relationship to humans can be described as commensalism, with crows choosing to live within 3 miles of humans. They can even remember human faces and pass down generational grudges. Crows have become intelligent witnesses to human lives, surviving as neighbors within the modern natural world.
Join me on a journey into a world mediated by tiny enzymes, biological codes, and a lesser-known mediator we call SNPs.
The problem is that when you look at mental health, everybody keeps his votes on the care. Now it is, at the end of the day, if the dopamine system is not working correctly, you're going to have mental health issues.
The mysterious, beautifully complicated world we call the brain is always within us, but somehow still untouchable. The brain and body are limitlessly complex, so let's break down some of the essentials. DNA is the genetic material within our cells that is made up of 2 long strands of paired nucleotides. Nucleotides are base pairs connected to the DNA backbone that bond together to form unique patterns to make up genes. Genes are the portions of DNA that encode the proteins that make up every functional unit in our bodies. Throughout time, humans have had mutations in their DNA that's gotten passed down and established in their community. These small mutations can have noticeable effects on the brain's functions, especially when paired with mutations that compound the effects of the first. But most people are not associating their quirky habit with a mutation in their DNA. While many habits can be attributed to nurture, we may be missing half of the equation that deserves just as much attention: the nature. These sometimes quirky mutations in single nucleotides are called single nucleotide polymorphisms, or SNPs for short. Any mutation that's been able to spread to at least 1% of the population is a SNP. We have many biological processes that are mediated by the expression of these SNPs within the brain. Almost all the differences that make you are mediated by SNPs. 99.9% of your DNA is the same as every other human you've met. The remaining 0.1% of genetic material is mostly made up of SNPs, which accounts for 90% of the differences between individuals. While most SNPs are inconsequential, some of them can change the way that we interact with the world. There's a chemical often associated with joy and feeling good that most people have heard of: dopamine. Dopamine is a chemical in the brain known as one of the most important neurotransmitters necessary for reward pathways, working memory, habit formation, motivation, and even motor control. Within the brain, we have receptors that catch the dopamine in order to feel its effects, and these receptor genes are no exception to being affected by SNPs. The DRD2 gene codes for the dopamine 2 receptor and contains many well-studied SNPs. But it is also co-expressed with other genes, such as the ANKK1 gene that contains the A1 allele associated with 40% less dopamine 2 receptor density. The brain requires receptors to accept the dopamine that is made, and having fewer receptors leads to a diminished ability for the brain to use this vital chemical.
The DRD2 gene integrates all mental health, so that's how important it is.
The limited neurotransmission of dopamine is linked to a not-so-unknown diagnosis: ADHD. The symptoms, such as hyperactivity and inattention that individuals with ADHD may have, are strongly associated with an imbalance of dopamine. Many individuals with dopamine imbalance have overlapping symptoms, but have completely different brain chemistry. Dopamine balance and homeostasis are the goals, so understanding the mechanisms behind these symptoms can help avoid catastrophic medication side effects. Studying SNPs opens the door for targeting the root of dopamine imbalances, from patients with ADHD to Parkinson's disease. You can unlock the secrets within your genes with a $30 SNP test without needing to test your whole genome. These tests can help explain confusing symptoms and encourage the correct path forward. The DRD2 gene plays a vital role in integrating the processes we need to maintain dopaminergic homeostasis. Our current genetic technology creates more opportunities to manipulate our gene expression with increasing precision, and genome testing gets cheaper every day. Linking SNPs to neurodevelopmental disorders such as ADHD is just the start of what our genes can tell us. Living in the era of information allows us to break down complex data like never before. Take your genes into your own hands. Do not suffer in silence. Your power lies within your knowledge. Living in a culture made for wild-type genes can make our SNPs feel extra heavy, but they don't make you broken. They make you human.
A groundbreaking new clinical trial out of the Fred Hutch Cancer Center is fundamentally changing how we approach stem cell transplants for severe blood cancers. Recently, published in the Journal of Clinical Oncology, the Phase 2 study investigates a new cellular product called DILANUBACEL. The goal? To make single-unit cord blood transplants faster and safer for patients battling blood diseases like leukemia. Leading the way is Dr. Milano, who is the first author of this groundbreaking paper.
Hi, I am Dr. Filippo Milano, and I'm one of the attending transplant physicians here at Fred Hutchinson Cancer Center at the University of Washington. My area of expertise is cord blood transplantation.
To understand why this research paper is such a massive leap forward, we first have to look at what cord blood transplantation is and what problem DILANUBACEL is trying to solve. When a baby is born, the umbilical cord leaves behind something remarkable: cord blood containing hematopoietic stem cells. You can think of these cells as a biological blank slate. They naturally reside in our bone marrow and have the incredible ability to renew and differentiate. And the best part? Collecting it is completely painless for the mother and child. For leukemia patients whose own bone marrow is failing, a stem cell transplant can literally replace their diseased immune system and save their life. But there is a catch. A single unit of cord blood does not contain a lot of stem cells. Because of this low cell count, it can take up to 3 to 5 weeks for the new immune system to grow and engraft. During this long waiting period, patients are highly vulnerable to life-threatening infections. The current solution to this problem is using 2 units of cord blood at once. But doubling up increases the risk of a dangerous complication called graft-versus-host disease, or GVHD. This is where the donor cells actually attack the patient's own body, causing many severe side effects, such as painful rashes. This is the exact problem Dr. Milano and his team set out to eliminate. They want to see if they could rely on a single unit of cord blood by pairing it with a new cellular product called DILANUBACEL. To get a better idea of this product, we spoke to Dr. Milano.
DILANUBACEL is a pooled product deriving from 6 to 8 cord blood units pulled together. This product has been developed here at Fred Hutch, where we started doing use of expansion technology for patients undergoing cord blood transplantation. How does that work? Basically, you get 6 to 8 units all together, you pull together in the same bucket, and then you do a selection of CD34, which are stem cells, and then you expand these cells for about 2 weeks. After these 2 weeks of expansion, you end up with multiple units containing different doses of stem cells, and you can just put them as an off-the-shelf unit ready to be used along with the other stem cell sources. In this study that was published in the Journal of Clinical Oncology, we pair DILANUBACEL, once again 6 to 8 cord blood units pulled together, to a single cord blood unit which was matching with the patient. We did that all 30 patients over a period of 3 years, and we obtained a very important outcome, so with the 96% survival and only 2 patients relapsing after transplant.
The clinical outcomes following this new approach were striking. Out of the 28 patients treated, there were 0 graft failures. Patients were able to recover their primary white blood cells needed to fight off bacterial infections in a median of just 18 days. Beyond the faster recovery, another massive takeaway from the study is the safety profile. Despite DILANUBACEL originating from multiple donors, researchers observed 0 cases of GVHD. A year later, 27 of the 28 patients remained alive and disease-free. By safely accelerating recovery without causing dangerous side effects, DILANUBACEL is paving the way for safer and faster transplants.
Now where we are with this, we need to go and move back from the bed to the bench, trying to understand how we saw so many good results using this approach. And we are also trying to test this approach with other stem cell sources, like the one coming from matched or mismatched already donors, and also from stem cell sources coming from family when we use haploid identical transplant.
To conclude, DILANUBACEL is made from a combination of 6 to 8 cord blood donors. It is administered just hours after a transplant for a cancer patient and acts like a booster shot, helping the new immune system grow back at a much faster rate. The contrast is remarkable. DILANUBACEL only requires 1 cord blood unit instead of the previous 2. Before DILANUBACEL, Using multiple cords meant a much higher chance of graft-versus-host disease, and cell growth took weeks. With this booster, patients safely bypassed that dangerous complication entirely, and their vital white blood cells recovered in less than 3 weeks. This proves DILANUBACEL is a product worth looking into.
In the distance walks the largest land animal on the planet, the African elephant. These gentle giants are not only known for their size, but also for their incredible intelligence, memory, and strong social bonds. With these skills, elephants have learned to adapt and survive in the harsh, unforgiving climate of the African savanna. Here we follow a herd beginning their search for water, which is not an easy task. Elephants walk up to 483 kilometers to find these sources, and it is incredibly difficult during times of drought. Leading this journey is the matriarch, who is named Jasiri. She is the oldest of the group who retains the knowledge from generations before her. Her experience and understanding of the land are essential for the survival of the group. The rest of the herd stays close around her and may maintain physical contact with one another to show their trust and connection. Mothers keep their calves within reach to ensure they are protected from any potential dangers. These interactions show the incredible emotional capabilities elephants have. There is power in groups, and they have found this cooperation to be essential for survival. As they continue their walk, they feel the temperature rising, making the search for water urgent. Elephants use water for evaporative cooling because they lack sweat glands. This helps them thermoregulate, especially in heat like this. On a normal day, elephants will drink around 50 gallons of water, which makes it essential that they reach a source soon. The herd has now found the watering hole, or at least what's left of it. Their once familiar and reliable water source is no more. The savanna is merciless. During the dry season, rainfall decreases, fresh crops become scarce, and watering holes go dry. The land offers no relief, and these elephants run the risk of heat exhaustion. Jasiri digs into the dry riverbed in hopes of finding a source of water underground, but in these extreme conditions, she has no luck. She must lead the herd to a different water source. In the meantime, she is walking them to where there is loose dirt and sand available. They then gather the sand and throw it on their bodies, providing them temporary protection from the sun. They also flap their ears to increase airflow over the blood vessels inside them. This helps heat dissipate into the air and cools the blood, which then circulates throughout the body and lowers the elephant's overall body temperature. While these efforts will provide some relief, it will not be enough. It is time for the matriarch to show her wisdom. Jasiri begins to lead the herd to another watering hole used by their ancestors. In this moment, her memory is a matter of life and death. The walk will be long, but this is their only option. After another brutal 80 kilometers, the herd reaches their destination. They are exhausted, but ready to get in the water. Bathing provides an immediate cooling effect, as well as traps water in their intricate wrinkles and crevices that then transfer heat into the air. This evaporative cooling is an elephant's version of sweating, and it is essential for their survival in these temperatures. After the elephants spend their much-needed time in the water, which is often an hour or more each day, they head over to the mud areas to cover their skin with this natural sunscreen before they begin on their next journey. Their survival does not come from their size or strength alone, but from intelligence, memory, and working together to ensure that no elephant gets left behind. Jasiri has fulfilled her role as matriarch by guiding her herd through every hardship utilizing the insight of her ancestors. She has secured the future of generations to come and is ready to pass her wisdom on.
Originally, I came out here thinking all seagulls look the same, but it turns out there's many different types of seagulls. Come along with me to learn about two different types, the kelp gull and the dolphin gull. Although both the dolphin and kelp gull reside in the coasts of the southern hemisphere, the kelp gull is found in the Antarctic, Patagonia, northern Argentina, and the Pacific coast. And the dolphin gull is found in the southern coast of South America, from Tierra del Fuego, north of Curaçao, Argentina, and then north to Chihuahua Islands. Their differences are also seen in their looks. When you think of the seagull, the kelp gull is probably what you think of, with yellowish legs, a white body and head, dark gray or black upper parts and wings, and a bright yellow beak. They also range between 0.53 to 0.66 meters, or 21 to 26 inches in length, and weigh about 453 to 1,360 grams. Whereas the dolphin gull is quite different, with red legs and beak, a dark gray body, and even darker upper parts and wings. They are also smaller than kelp gulls, ranging from 0.381 to 0.457 meters, or 15 to 18 inches in length, and weigh about 521.6 grams. So clearly, the dolphin gull has a smaller build than the kelp gulls, but this is mainly due to their feeding habits. Kelp gulls are often described as opportunist generalists because they're omnivores and they're able to eat different forms of food. Their diet includes mainly mollusks, fish, worms, amphibians, small mammals, other birds, and even their eggs. But for the dolphin gull, it is even more interesting, as they like to play the waiting game. They mainly feed off the colonies of the southern sea lion and Magellanic penguins. Oftentimes, when Magellanic penguins feed their chick, they usually put the food directly into their chick's mouth with their beaks. The dolphin gull would attempt to distract the parent giving food by poking their behind or areas they cannot see. making the penguin turn, drop the food, and then the dolphin gull will be able to get the food once the penguin leaves. And during September to February, their source of food stems from sea lion excrement that they release on the shore. It's very obvious that the dolphin gull is a lot more passive in getting their food than the kelp gull. Unlike kelp gulls, who would actively seek for other birds' eggs, there are no records of dolphin gulls ever stealing eggs. Because of their docile feeding strategy, dolphin gulls are often displaced by skuas, snowy sheathbills, and even kelp gulls. Because of this and their reproductive behaviors, dolphin gulls are often seen in groups. Their colonies can range between 2 to 210 pairs, but are oftentimes less than 25 pairs in Punta Tambo. In comparison to the kelp gulls, who forage individually or in small groups, as they are opportunist scavengers. But even with all the differences between the kelp and the dolphin gull, they have one thing in common: the avian flu. This is to the kelp gulls in particular, since they have more variety in their diet, making them even more susceptible. Symptoms being swollen head, closed running eyes, unresponsiveness, drooping wings, twisting, bleeding, and redness of the legs and neck. A specific strain of the avian flu, called H5N1, can be passed on to humans through contact of infected wildlife. Although it is rare, it is important to be cautious when interacting with birds, like the kelp gull, who are more vulnerable. When I first started this project, I honestly thought a seagull was just a seagull. But after learning about these two species, it's clear that every animal has its own behavior, role, and story within its ecosystem. So next time you see a gull flying overhead, take a moment to look. You never know what story is hidden in plain sight. Thank you for joining me today. My name is Kimberly, and I hope to see you in my next journey. Whether it's a dolphin gull living along the coast with their colony, or a kelp gull surviving out of their own, these birds show how wildlife constantly adapts to the changing environments.
Scientists have relied on the written word as a means of relaying information for thousands of years. Over the years, the written word has improved to make the spread of research much easier. Due to recent developments in technology, however, science communicators are using different ways to relay information to reach new audiences. Literacy for US adults is not a given. 54% of adults read below a 6th grade reading level, and 21% of adults are considered illiterate. Reading specialized scientific papers is already difficult for academics outside of their specific field, so reading scientific papers is nearly impossible for the average American. Science communicators use video to create digestible media for everybody to enjoy.
Hi, I'm Christian Ekstrom, and today I'll be guiding you through the process of creating digital scientific media. Together, we will focus on an old project of mine that analyzes chronic wasting disease, a disease that affects members of the deer family.
The creation of every science video begins with a simple interest, followed by lots and lots of research. The interest of the project, as previously mentioned, is chronic wasting disease, a disease found in cervids, or members of the deer family, that attacks the nervous system. 1 in 5 Americans hunt deer, and 3 in 5 have eaten deer in the past year. Cervid health must be closely monitored due to human consumption. Chronic wasting disease, or CWD, is a fatal disease that affects all members of the deer family caused by misfolded proteins called prions. All proteins are supposed to be folded a certain way, either as a beta sheet or an alpha helix. Prions are proteins that do not conform to their destined shape. Humans can contract a similar prion disease called Creutzfeldt-Jakob disease. This disease acts in the same way, by infecting the brain and attacking the nervous system with no hope of survival.
Now that our topic has been identified and thoroughly studied, we must collect our footage. The footage for this project was collected on trail cameras and from submissions by local hunters. These trail cameras have low light capabilities, which is ideal because deer are crepuscular creatures. Crepuscular means that deer are most active during dawn and dusk.
Once an abundance of deer footage has been collected and analyzed, the footage will be fused together to create a cohesive visual aid. This process can take several hours, or even days, depending on the length of the film. Time is an important factor, as the story must be concise, so only clips that further the story will be used. The rest will be left out. Chronic wasting disease is spread through close contact and exchange of bodily fluids with infected animals. These bodily fluids include saliva, blood, urine, genetic material, and antler velvet, so videos of cervids in close proximity are most beneficial. There are no visible symptoms of chronic wasting disease during the majority of a deer's infection, a time that spans 18 to 24 months, with the symptoms of weight loss, behavioral differences, and lower muscular control only showing shortly before death.
Since the window is so short for me to collect footage of deer with chronic wasting disease symptoms, I've got to rely more heavily on graphs and illustrations for visual aids.
After all of the visuals are made for the project, the audio is created. A script is read and pieced together.
Deer, elk, and moose. Natural science videos rely heavily on natural sounds. Unfortunately, not all of the footage that we use has acceptable audio that we can put into our final product. To combat this, science communicators use a technique called Foley, where we recreate the audio ourselves and then we overlap it onto the existing footage. Since cervids typically live in wooded areas, I've decided to recreate some of the sounds most commonly heard in these wooded areas. These sounds include the breaking of branches and twigs, and also the rustling of grass.
Once the noises are added, the work is ready for a soft launch.
So I think the foundation is good. The audio could use a little bit of work. It's kind of gargly in some spots. The background, like noise from the actual clips themselves, hasn't been removed. It could use some background music. In some areas, the camera work is also a little bit shaky, but I think it's a good start.
After getting feedback from small groups of trusted people, it is time to go back to editing, changing and refining the content. This process will continue until the product is finally polished into an acceptable work.
Once the product has been refined, it'll be uploaded to the internet for the world to see. And thanks to our efforts, science becomes just a little bit more accessible to the average Joe.
Saliva. You make about 1.5 liters a day, and most of the time, you don't even notice it's there. Until this happens. Think about your favorite food. Fresh pizza, warm cookies, sour candy, and suddenly, your mouth starts watering. Let's think about this. How can just the thought of food trigger tiny glands in our mouths to start secreting saliva before we even take that first bite? To answer that, we need to think of how is saliva secreted, what is saliva actually made of, and why do we secrete it in the first place? We can break down the digestive system into three major parts. This includes the cephalic phase, the gastric phase, and the intestinal phase. Saliva secretion is in the cephalic phase, so we'll focus on that. The cephalic phase introduces four main digestive processes: secretion, absorption, digestion, and motility. Saliva contributes to all processes, chemically breaking down carbohydrates and physically breaking down food, creating boluses, in order to travel to the gastric phase. In the cephalic phase, we also have a natural adapter reflex. This is known as the feed-forward reflex. With the anticipation of the ingestion of food, we naturally activate housekeeping processes, such as secreting saliva to help digest food. Saliva is secreted by three main salivary glands: the submandibular gland, the sublingual gland, and the parotid gland, this one being the most dominant and an exocrine gland. Looking at the parotid gland, we can dive deeper into the cells within it and see what actually drives saliva secretion. In this case, it's osmosis. which is where water molecules follow where the most dissolved particles are over a membrane to reach equilibrium, which then this creates saliva secretion. But again, how are we doing this so easily? It's not like we're thinking about the production of saliva. Usually we're just thinking about how good the food's going to be or how good it is. The production of saliva is a long reflex and a positive feedback loop. So signals are sent through our central nervous system to our brain, amplifying a response to our stimuli. Think of a dog when it sees its owner refilling its bowl for dinner. The dog starts to salivate, preparing to eat food due to the signals registered from the optic sensory nerve site. Seems simple, but it's actually super complex, and this is why we must appreciate it. A simple control model is nice to break it down. A control model includes a stimulus, sensor, input signal, integrative system, output signal, target, and response. For an example that applies more to humans, let's imagine you're walking down Pike Place and you smell donuts. This smell acts as your stimuli, which then is received by your olfactory receptor, which is specifically for smell. Then action potentials are sent down input cranial nerves, which then is received by your integrative center. In this case, we are talking about a long reflex, so your brain. And that sends output signals into your target, which is going to be a parotid gland for our example, which then causes the response of salivary glands to secrete into your oral cavity. We can also look at this process through physiology. Let's give credit to ourselves and get into the nitty-gritty. Salivary glands resemble a bushy-like structure. The grape-like ends are known as acini, which is connected to the duct, which then eventually leads out to the oral cavity. The acini ends have acinar cells, while the duct has ductal cells. Now, when thinking about the Pike Place donut, acinar cells receive that signal and begin the process of saliva secretion. They produce an isotonic solution, meaning the amount of ions in water are balanced. This happens by drawing the ions potassium, sodium, and chloride by the process of flux, where ions are drawn into the cell by either an electrical or concentration gradient. The same process happens when being pushed from the intracellular fluid to the lumen, apical side of the cell. The channels allowing ions to move from the ECF to the ICF are known as membrane proteins, each having their own role. For simplicity, we'll look at the movement of chloride. The MKCC facilitated diffusion protein actively draws chloride into the cell using ATP, opposite of chloride flux, due to the charge of chloride repelling the membrane potential charge, as well as the concentration being lower on the ECF. Since the resistance is higher in flux, the ion must be actively pushed into the cell. When in the cell, the repelling of charges causes chloride to be passively pushed out of the cell into the lumen through a chloride channel. Once this ion has reached the lumen, water fluxes through aquaporin channels, allowing water to move through the cell. It also moves through tight, leaky junctions between the acinar cells, which allows water to move around the cells. The water fluxes through due to the drive of equilibrium, diluting the high concentration of ions in the lumen. This creates a tonicity, volume of the cell, known as isotonic, where the level of ions is balanced to the water. The buildup of isotonic solution allows there to be a greater pressure, moving the saliva production down the duct, which then interacts with the ductal cells. And with their certain tight junctions, they only take up the ions, allowing more water to be within the duct, causing a hypotonic solution, and then that is secreted into the oral cavity. Like I said, compared to the acinar cells, the ductal cells have tight junctions, which don't allow water to pass through and leave the lumen. But they have exchangers and channels for ions to be taken out of the solution. Chloride is exchanged with bicarbonate and ions such as sodium that are taken up and put back into the ECF through the same driving gradients of concentration and charge, to be recycled for the acinar cells when stimulated again. This hypotonic solution now contains bicarbonate, water, mucus, and enzymes, which now will be secreted into the oral cavity, allowing us to chemically and mechanically break down food. We truly can never forget how fundamental saliva is for our overall health. As mentioned, saliva lubricates boluses, allowing it to move to the next phase of the digestive process, that is, the gastric phase. And at the same time, the solution has enzymes, which breaks down carbohydrates. And additionally, a little fun fact is it also helps us against pathogens, having antibacterial properties. Personally, I think the most important function of saliva is enhancing flavors, dissolving particles in our mouths, allowing our brains to recognize certain tastes. And speaking of which, this apple is making me salivate right now, which is kind of funny because a few moments ago, I wasn't even thinking about saliva, but now it's the reason I'm ready to take my next bite.
For humans, beaches are synonymous with vacation and relaxation. It's where many people go to enjoy leisurely activities on their days off. Washington State has over 5,000 statute miles of shoreline, making it home to plenty of beaches, some of which are part of major cities in the region. Many of these beaches and big cities are located in the Puget Sound, an inland estuary connecting to the Pacific Ocean. A core component of beaches is the intertidal zone, which is where the ocean and land intersect. There are 4 divisions of the intertidal zone, but what they all have in common is that they transition from periods of being submerged to periods of being exposed. The back and forth conditions that are characteristic of the intertidal zone can pose great challenges like desiccation and internal fluid regulation, contrasting the calm vibes many envision. The creatures that inhabit this space must be able to function in highly dynamic environments that change 4 times per day. But what species are able to not just survive, but thrive here? 2 native shore crab species dominate the rocky beaches of the Puget Sound: the purple shore crab, or Hemicrapsis nudus, and the yellow shore crab, also known as Hemicrapsis oregonensis. They occupy the higher and lower intertidal zones respectively, although their territories can overlap slightly. Let's take a close look at these crabs to see how they have adapted to the intertidal zone. One of the most dangerous factors of the intertidal zone is the rising and falling of the tides. The movement of water channeling over, under, and around the rocky shore can create rapid flows that sweep all sorts of material out to sea. Shore crabs are particularly vulnerable due to their size. Purple shore crabs have shells that average a width of 5.7 centimeters, while yellow shore crabs are even tinier, measuring 5.1 centimeters. Consequently, their tiny bodies feel the forces of the waves even stronger than others. However, these Hemicrapsis species have evolved behavioral traits that promote their survival. Shore crabs wedge themselves underneath rocks to avoid exposure to the currents. The rocks provide sufficient protection and shelter for the shore crabs, as they are heavy enough to stay in place when the water swirls around and crashes down onto the shore. The behavioral instinct to seek out shelter also helps the crabs conserve more energy, as the rocks protect them from most of the brunt force. Conversely, there is also a real danger to staying away from the water. In the northeastern Pacific, where the Puget Sound is located, the high intertidal zone experiences some of the longest exposure times to air. In the winter, the lowest tide occurs at night, and crabs are prone to being frozen. But in the warmer months, the lowest tide happens during the day. The combination of sunlight and the lack of water can desiccate shore crabs, especially the purple shore crabs that are found higher up in the intertidal zone. Desiccation is a major threat, especially when the water has receded. To keep themselves from drying out, purple shore crabs will scurry down to the water. This keeps their moisture levels high enough to live and also helps with thermal regulation. To keep their body temperature relatively constant, they will scuttle in and out of the water to avoid overheating during low tide. Additionally, as an added evolutionary safety feature, purple shore crabs can tolerate immersion for over 24 hours. While yellow shore crabs can worry less about desiccation since they are submerged for longer periods of time, they face a new survival challenge: salinity levels. In the intertidal zone, the salinity levels of the water can vary drastically, and only fully aquatic crabs like cancer crabs can survive in water deep enough to escape the variation. If it rains, salinity levels decrease as the ocean water gets diluted with the rainfall. However, when the water near the shore evaporates, the salinity levels can double that of the deep ocean. Yellow shore crabs are able to tolerate the fluctuating salt concentrations for extended lengths of time, especially when salinity levels increase. They do this through hypoosmotic regulation, a process where organisms keep their internal body fluids at lower salt concentrations than the outside environment. Hypoosmotic regulation allows yellow shore crabs to survive in salinity levels that vary up to 180% of the average ocean salinity. There are other hurdles for these shore crabs to navigate, such as surviving predation and competition from invasive species. But even just looking at a few key environmental challenges reveals a huge picture about the lives of these minuscule creatures. Shore crabs, despite being so common in the Puget Sound, are tiny and unassuming to many, including researchers. It could be because other crabs are better for human consumption, or because shore crabs are overshadowed by marine keystone species. But regardless, not much is known about them yet. Their challenges can also appear minor to larger creatures like humans, but it is worth it to zoom in and give the native Hemicrapsis crabs a good, close look, because their characteristics can teach one extensively about the inconspicuous yet volatile nature of the intertidal zone.
This is Husarik, a majestic ruler of the African savanna. To us, he looks like an untouchable king, commanding an empire with absolute power. But a king is nothing without his kingdom, and for a lion, survival is a shared burden. Within Husarik's pride, bonds are forged through touch. This constant grooming amongst his lionesses isn't just about cleanliness, it reinforces the social structure that keeps his kingdom unified. This sisterhood is the bedrock of his empire. These females are all closely related, sharing an unbreakable lineage that Husarik is charged with protecting. Together, they raise his offspring communally. The mothers nurse and protect each other's cubs, ensuring the collective future of Husarik's bloodline survives. But protection is only the first step. His cubs must eventually learn to face a harsh, changing environment. Hunting is not an innate instinct born overnight. It is a highly complex, coordinated skill developed slowly over time. Through playful wrestling and mock stalking, these young cubs are practicing the crucial coordination they will need as future apex predators. But today, the upcoming hunt is far too dangerous for these young ones. The target is large, unpredictable, and fiercely defensive. One of his primary lionesses leads his cubs back to a safe distance, away from the impending chaos of the tracking line. With his cubs hidden, the adult hunting party moves out in a perfect silent unison, fanning out into strategic positions across the plain. Their targets are migrating herds. Hundreds of wild beasts and zebras moving across the terrain, representing a vital source of life. The pride tracks them to the water's edge, waiting for the perfect moment of vulnerability to strike. In a burst of dust and raw power, the trap is sprung. A single successful takedown secures the pride's future. The hunt is over, but the work isn't. Lions can spend up to 4 days guarding and eating a single large meal, consuming everything to sustain their massive energy demands. They eat and rest under the cover of darkness, but it's never quiet for long. From the depths of the savannah, a challenge emerges. A coalition of nomadic males roaming the plains. They are young, hungry, and seeking an empire of their own. Husarik's kingdom is no longer safe. His duty has to shift from protector to guardian of his bloodline. The pride senses this shift and heads towards the commotion. The clash is inevitable. When the nomads finally breach his territory, the savannah erupts in a violent struggle for dominance. The cost of defeat is absolute. The victorious males target the pride's youngest. By removing the existing cubs, they force the lionesses back into a breeding cycle, ensuring that any future lineage only belongs to them. In the silence of the aftermath, Hodorai's world has been shattered. The pride he defended, the family that kept him a king, lost to the raw brutality of the wild. Now he walks the plains as a ghost, a deposed ruler of a broken empire, the ultimate cost of being king.